look at my PowerPoint?
Professor, no, I
Oh,
only
oh, I see. Okay,
I
sorry,
yeah,
sorry.
sorry, Professor. Uh, I only let you.
Uh, okay, just a second. Share. Window. Yeah, should be.
I can look your slides, Professor.
Okay, thank you very much.
Thank you, Professor.
Okay, okay, let's start the course number 3. Sake Advance. Actually, this is the photo I took yesterday. Big tree fall down and our deck totally broke. About half of deck broken. So because yesterday we had severe weather, window, and big tree fall down. It destroyed my house. When I was making a class through online to the primary kids, student with the university in Japan, Ochanomizu and Tokyo Gakugei Daigaku University, we have been working together. And during my online class, the big tree fall down. Anyway, let's start talk about this is a subject, alcohol fermentation and alcohol molecules. So first, about half first part of this session, I just review the previous lecture about the respiration and fermentation. And I will move on to alcohol fermentation and especially bad effect of alcohol on our health. This afternoon, I will focus on the not good side of alcohol molecule. Okay, this is the schematic diagram showing mitochondria. You may have heard the name of mitochondria, which is an organelle within cell. It has two membranes, bilayer membrane, outer membrane, and inner membrane. Okay. And as I told you last week, glucose will be converted to two molecules of pyruvate and within cytoplasm. And the pyruvate move into inside of the mitochondria. And it will be degraded to CO2 and H2O, water, through citric acid cycle in the presence of oxygen. So this is respiration. So respiration occurred in two sites, two locations. One, glycolysis occurred in cytoplasm, and citric acid cycle, or TCA cycle, will be performed within mitochondria in the presence of oxygen. If we don't have enough oxygen, in the absence of oxygen, pyruvate, two molecules of pyruvate are the last compound. It will not be degraded further to CO2 and water inside of mitochondria. So pyruvate will not go to inside of mitochondria in the absence of oxygen. But in the presence of oxygen, pyruvate go to inside of the mitochondria. So this is outer layer of mitochondria. This is inner membrane of mitochondria. Pyruvate will be degraded. Pyruvate broke down further to CO2 and water within mitochondria. And electron and proton will be moved to the space between outer membrane and inner membrane. So you can see H plus, this is proton, and also electron move together from inside matrix of mitochondria to the area, intermembrane space between outer membrane and inner membrane. So electron, proton will be pumped out from here to here. It makes the gradient of concentration of electron or proton. So concentration of proton or electron in intermembrane space will be higher than it in matrix because electron and the proton pump out will be pumped out from here to there. So this is just like a dam. So water will be pumped up to the inside of the dam. So this area has more proton and electron compared with it in matrix. And last week I told you that this is a kind of turbine, ATPase or ATP synthase, which is an enzyme to produce ATP. So this is the ATP synthase or ATPase. So electron and proton penetrate inside of ATP synthase. And ADP and inorganic phosphate makes ATP. So this part, which side? This is the inside. So this is the inside. So electron will, move from here to there, from intermembrane space, to matrix. From here to here. So from here to here, electron and proton. So this is the area of the membrane. So this part are located inside of the membrane. And when 3 proton or 3 electron move from here to there, so each electron rotate 120 degrees of this part. And 3 electrons moved, 1 molecule of ATP will be produced. So this is the respiration. So this occurred inside of the mitochondria. So 3 electrons move from here to here. So each electron rotates 120 degrees of ATP synthase. And 3 electron movement produce 1 molecule of ATP. And total 36 molecules of ATP will be generated from 2 molecules of pyruvate, from 1 molecule of glucose. So again, this is the schematic diagram showing membrane of mitochondria. This is the intermembrane space. So we have another membrane here, and this is the inner membrane. We have outer membrane here. So between 2 membranes of mitochondria, electron or proton accumulated from inside to outside. This makes gradient of concentration of electron between intermembrane space and matrix. So just like water, water flow from higher place to lower place. So just like that, when we open this pore, electron or proton move from here to here, from outside to inside, and it makes rotation of turbine of ATPase enzyme, which is a protein, catalyze ATP production by utilizing ADP, adenosine diphosphate, and inorganic phosphate to make adenosine triphosphate, adenosine PPP, 3-phosphate attached to the adenosine. And 36 ATP will be produced from 2 molecules of pyruvate, from one molecule of glucose. So it means 36 ATP. In order to make 36 molecules of ATP, 36 by 3, 108 proton, 108 electron move from outside to the inside for generating 36 molecules of ATP. So this is respiration. So this shows the protein chain to transfer electron from inside to outside. So electron will be transferred from several proteins. So they pass electron, just like that, including cytochrome. Yeah, these are cytochrome. So these protein delay carry electron from inside to the outside. And this pore will capture the electron from outside to inside to produce ATP. So this is the respiration. This is a mitochondria. Kenny, can you turn on this movie, or maybe I should send the link to you through email?
Don't worry, Professor.
Okay.
Give me a moment.
Okay.
Only I need the name. Give me a little moment, Professor, please.
Okay, thank you. So this is materials provided by Harvard University. They have many good materials. And this shows how mitochondria works. This is one kind of organelle inside of the cell. So it has bi-membrane. It has two membranes, outer membrane and inner membrane. Should I turn off the file sharing?
Okay, Professor. I shared my computer.
Did you find? Oh, thank you very much.
Yes, I share my screen. Give me a moment.
Thank you.
I press on. Yeah.
Oh yeah, this is the one.
Yeah.
So this is mitochondria. Okay, thank you.
Thank you, Professor.
Mitochondria are organelles found in you.
Is okay, Professor?
Yeah.
Thank you, Professor.
Eukaryotic cells that play an important role in the production of ATP, the universal energy currency used in all cells. Most mitochondrial proteins are transported from the cytosol into mitochondria through specialized protein translocator complexes. Interactions between these complexes bring the outer and inner mitochondrial membranes into close proximity. The successful targeting of many proteins that reside in the intermembrane space requires translocator complexes that cross both the outer and inner membranes. After entering the inner membrane translocator, many of these proteins do not completely cross the membrane, but instead are released into the membrane and diffuse laterally. They then remain embedded in the inner membrane or are cleaved, releasing a portion into the intermembrane space. Very few resident proteins are simply transported across the outer membrane translocator complex. Matrix proteins do not transit through the intermembrane space, but are directly transported from the cytoplasm across the interacting outer and inner membrane translocators. The protein-rich matrix contains enzymes necessary for cellular respiration, a process during which carbon fuel molecules are oxidized and reduced electron carriers are produced. Invaginations of the inner membrane, called cristae, contain 4 large protein complexes that harvest electrons from these carriers. Complex 2 accepts pairs of electrons from succinate and transfers them through a series of redox centers to coenzyme Q. The lipid-soluble coenzyme Q is reduced by electrons harvested from complexes 1 and 2 and then diffuses through the inner membrane, transferring its electrons to complex 3. In complex 3, electrons are transferred through 2 distinct series of redox centers, which allow them to cross the inner membrane one at a time. Finally, electrons are accepted by cytochrome C, which carries them to complex 4, where they are transferred through another series of redox centers to their final acceptor, a molecule of oxygen, that combines with hydrogen ions to form water. Electron transport in complexes 2, 3, and 4 is coupled with the pumping of protons from the matrix to the intermembrane space. The resulting electrochemical gradient across the inner membrane is called the proton motive force. Protons flow back into the matrix through a component of the ATP synthase called F0. The membrane-embedded ring structure of F0 binds protons in the intermembrane space and releases them on the other side of the inner membrane into the matrix. Proton flow drives the rotation of the ring structure, which in turn leads to the rotation of the F0 central shaft. The rotating shaft sequentially contacts the 3 catalytic subunits of the ATP synthase F1 complex, altering the subunit's affinity for ATP and ADP, and catalyzing the synthesis and subsequent release of ATP. Most of the ATP synthesized in the mitochondrial matrix is consumed outside of the mitochondria. But the inner membrane is impermeable to ATP and ADP. Therefore, an ATP-ADP carrier is responsible for the coordinated import of ADP and the export of ATP across the inner membrane. In cells, mitochondria are distributed near sites where ATP and other mitochondrial metabolites are in high demand. Mitochondria move along cytoskeletal structures such as microtubules and can undergo dynamic changes in shape, including both fusion with other mitochondria and division. Perturbations in the dynamic behavior of mitochondria and alterations in mitochondrial membrane permeability are associated with the early stages of programmed cell death.
Okay, thank you very much.
Thank you, Professor.
Okay, thank you. So share my file. Window. So you had image much better than this slide. You have good image by the materials from Harvard University. So four protein complex are located within the membrane, and the electron will be transferred by utilizing coenzyme Q, and also cytochrome oxidase. And proton and electron penetrated or moved from inside to outside through these four protein complexes. And the gradient of concentration of electron higher outside compared with inside makes rotation of ATP synthesis or ATPase to make ATP. And this is the major part of respiration. But if we don't have enough oxygen, this will not be, performed, because we need to have oxygen to capture H2 to make water. If we don't have enough oxygen, this electron flow will not perform. So fermentation. So now you have more idea about respiration. If we have enough oxygen, glucose will be degraded into two molecules of pyruvate. And if we have enough oxygen, pyruvate go to inside of the mitochondria, and it will be broken down to CO2 and H2O through TCA cycle within matrix of mitochondria. And the electron, somewhere we have a proton, will be released from inside matrix to the intermembrane space of mitochondria. It makes ATP. 36 molecules of ATP will be made. Only 2 molecules of ATP will be made through this glycolysis pathway. And if we don't have enough oxygen, so this reaction will be stopped around the pyruvate. Further reaction will not be occur. And it depends on if the organism have certain enzyme, pyruvate will be converted to acetaldehyde and ethanol. If some organism has enzyme to make lactate from pyruvate, it is called lactic acid bacteria. So this is the pathway or metabolic pathway of ethanol production. In the absence of oxygen, pyruvate will be converted to acetaldehyde and ethanol. No ATP, no further ATP production will be made after pyruvate. Only 2 molecules of ATP will be made. It will be made through ethanol fermentation. So this is the ethanol metabolism of ethanol production or ethanol fermentation. These are 2 enzymes involved to make ethanol or ethanol fermentation. This is the pyruvate. So pyruvate will be produced from glucose through glycolysis pathway. The pyruvate produced from glucose through glycolysis pathway. And further modification will be made by two enzymes. Pyruvate will be converted to acetaldehyde. So this is acetaldehyde, very small compound, plus CO2. This reaction will be catalyzed by enzyme called pyruvate decarboxylase. The pyruvate decarboxylase produce acetaldehyde from pyruvate. And if organism has alcohol dehydrogenase, this is the name of enzyme, acetaldehyde will be converted to ethanol. So in order to make ethanol fermentation, two enzymes will be required, pyruvate decarboxylase and alcohol dehydrogenase. So yeast has both enzymes. And yeast can perform ethanol fermentation. So this is the how ethanol will be produced by ethanol fermentation in the absence of oxygen. So now you have an idea how respiration will be made in the presence of oxygen, how ethanol fermentation will be made in the absence of oxygen. So this is in the absence of oxygen. So most of ethanol, alcohol beverage with ethanol are made by yeast. Yeast has both enzymes. So from next slide, I will a little talk about the alcohol molecule. Some aspect of the alcohol molecule, especially focus on not so good things. So the later, most of this course, I will focus on the excellent or wonderful aspect of alcohol. But this afternoon, I will discuss about some issue which is not very good on alcohol molecule. Okay, this is a symptom of alcohol intoxication. This is so from this slide, most of materials, Duke University in I think North Carolina, state of North Carolina, they have good university, and Duke University is one of the good university. Most of materials are referred by their materials from Duke University, and this is also referred from this site of their materials. So this showed how more alcohol makes bad thing. So this is the worst scenario. If we took lots of alcohol in short period, the worst case, we may die. But if we stop to drink alcohol at a certain level, it will make good effect on our maybe we can relax with small amount of alcohol. But if we have more, and these are symptoms, excitation, not very bad. but poor judgment, impaired thinking, memory loss. This is not very good. So if we take more alcohol unsteadily gait, we cannot walk smoothly. If we will take more nausea, vomiting, sedation, and if we take more pass out, so we need to be careful how much alcohol we can take, but we should stop to drinking. Reduced breathing, so this is very bad. Cardiovascular collapse, so close to death. So if we take lots of alcohol in short period, our life will be jeopardized. So we have to be careful how much alcohol we can take, what we should take. And I will talk some more aspect what alcohol affect on our body. So I also try to put slide on the Google Drive so you can read carefully and you will have more idea from this slide. This is a schematic diagram showing dissect of our brain in front, back. So this area excitation. So usually we suppress or we inhibit bad behavior in the public. So it says suppress or inhibit behavior that are socially inappropriate and in person. But if we take alcohol, so this brake will be released and we may make some bad behavior. We feel everything will be all right, and sometimes we make bad behavior in the public. If alcohol will affect this area. Number 2, pleasure. So this area control our pleasure. So alcohol may not affect very much in this area. But sometimes if we have too much alcohol will affect taking lots of alcohol affect this area and we cannot stop drinking. So this is a problem by alcohol on this area. And number 3, loss of judgment. Front cortex. Again, if we had more alcohol and we cannot judge very well because of the problem on this area. And if we take more alcohol, next, this area will be affected. And our movement, such as touch or vision, and we cannot move very well also. Sedation, loss of consciousness will be occur if we have problem by alcohol in this area of brain. And respiratory depression will be made if this area will be affected by alcohol. So you can see by taking alcohol more and more, maybe first this area will be affected. Next, this area will be affected. And this will be affected if we have lots of alcohol. So alcohol will affect our nervous system, especially in the brain. How the alcohol will reach at the brain or cells within brain. So next few slides, I will explain how alcohol move to the cells within brain. Usually, drug will not be reached to the cell of brain, but alcohol can reach to cells in brain. So alcohol will be taken from the gut in our body and go to capillary blood vessel from intestine. And alcohol molecule, this yellow small, these are alcohol, will be absorbed by epithelial cell interstitial space and move to blood vessel, capillary, and move around in our body. About 30%, some alcohol will be absorbed, will be taken up by stomach, but most of alcohol will be taken by intestine and move to blood vessel, capillary. This is the endothelial cell. Non-brain capillary and brain capillary. This figure shows the difference, the structure of capillary or characters of capillary. Non-brain and brain capillary. Non-brain, like other area, like stomach or hand or foot. Charged molecule, or plus or minus, charged. A molecule will be moved from inside of the capillary to the outside of the capillary to mean the cell or tissue or organ from blood vessel to tissues or organs. So these charged molecule, negative charge, either negative charge or positive charge, charged molecule can move through a membrane of capillary cell or blood vessel cell. But the brain, in the brain, charged molecules cannot penetrate through a membrane of capillary cell or endothelial cell. Charged molecule, including drugs, stay inside of the capillary in brain. But alcohol, ethanol, or other lipophilic drug can penetrate, can move from inside to outside. So this is just like non-brain capillary. Non-brain capillary charged drugs as well as ethanol or other lipophilic drugs can move from inside to outside. But in the brain capillary, only ethanol or lipophilic drug can move through membrane of endothelial cell. Charged drug or charged molecule stay inside. So we can protect our brain from some bad molecule because brain capillary will not move the charged molecule from inside to outside. So brain can be protected by cells of brain capillary or cells, endothelial cells within the brain. but alcohol can go to the cells within the brain. So alcohol will affect the cells of brain. So each region has their own cells. And blood vessel, alcohol released from capillary blood vessel will affect the function of cell in the brain. So this is the difference between the character of endothelial cells, non-brain capillary, and the brain capillary. This figure also shows this is the alcohol molecule. This is a water molecule. So this is a membrane. So by a lipid bilayer is our membrane, and some protein locate within membrane. Lipid bilayer. This each circle with two lipid molecule. The water can move only through the protein. So protein with charged location by using charged area within protein, because water has plus and minus charged molecule. Water is a charged molecule. So water cannot penetrate lipid. So water and lipid will not be mixed. So water only can move through protein area. But these are alcohol. Ethanol molecule can move through protein as well as by lipid bilayer or membrane. So ethanol can penetrate membrane just as I showed, just like endothelial cell within brain. Ethanol molecule can penetrate because of its character. And even in the brain, easily ethanol will penetrate to inside of the cell of brain. So next few slides. So now you know that the ethanol will reach the cells within the brain. So next few slides, I will explain how alcohol will affect our health. This slide shows alcohol will be breakdown, will be degraded by two enzymes. Eventually, alcohol will be degraded to water and CO2, and we just discard. In order to make alcohol into CO2 and water, we need to have two enzymes. One is called alcohol dehydrogenase. Another one is called aldehyde dehydrogenase. So two types of dehydrogenase will be utilized, will be required to degrade alcohol in our body. So this is the mechanism how to break down the ethanol. This is the ethanol molecule. This is a cytoplasm. Here is a mitochondria. So inside of the cell, outside of the cell. So ethanol will be transferred, moved to the cell. Ethanol will be released from capillary. Ethanol will be released from capillary around in your body. And ethanol will enter each cell next to capillary. And it will be degraded to CO2 and H2O through two steps. One is catalyzed by alcohol dehydrogenase, convert to ethanol to acetaldehyde. This is very toxic. I will explain how this molecule are toxic in the later. Acetaldehyde. And next, acetaldehyde will go to inside of the mitochondria. And in the mitochondria, acetaldehyde will be converted to acetic acid. This is not very bad. Acetic acid has no toxicity. So we need to convert from ethanol to acetaldehyde quickly, and we should be okay. But some people, it takes long time to convert from ethanol to acetic acid, and they will have problem. They feel sick. So we need to have two enzymes. This is the name of enzyme, aldehyde dehydrogenase. Will convert from acetaldehyde to acetic acid. So these two enzymes are required to have to make ethanol to acetic aldehyde, which is not toxic compound. So this is one slide showing usually female tend to have higher concentration of alcohol because of the water content. In general, water content of female has less than that compared with male. Male has 68% of water inside of their body. And average female has 50% weight come from the water. So if they take same amount of alcohol, and female tend to have higher concentration of alcohol because they do not have much water. So they cannot dilute alcohol efficiently compared with male does. This is another comparison between male and female. Male, female. So this green dot showing ethanol. When we take ethanol, it goes to the stomach first. Next, it will be moved to liver. Male can convert ethanol to acetaldehyde in the stomach. Not all, but some ethanol can be converted to acetaldehyde within stomach in the case of male. Female has an enzyme. Female has a gene for, what is the name of alcohol dehydrogenase. Both male and female cells of the stomach has gene for alcohol dehydrogenase, but for some reason, female does not convert ethanol to acetaldehyde in stomach. And if alcohol move forward to liver, and the function of the liver of male are very strong. So the enzyme activity of alcohol dehydrogenase within liver in male has more activity compared with female. In general, it depends on some, male does not have much activity, but some female has very strong activity, may have in the liver. But in general, male can convert ethanol to acetaldehyde efficiently, more efficiently compared with female. So due to these enzyme activity or allocation of enzyme expression or amount of water, in general, male tend to convert efficiently to ethanol, from ethanol to acetaldehyde efficiently compared with female. So this is just a review how ethanol will be degraded to acetate, which is not toxic compound. So two enzymes, alcohol dehydrogenase, male can have some activity of this enzyme even in stomach. The female does not have. Even female has gene of this enzyme in the cell of stomach. But activity, this enzyme convert ethanol to acetaldehyde. And alcohol, and another enzyme is required. Another enzyme called aldehyde dehydrogenase. Next is required to convert acetaldehyde to acetate, ALDH. So two enzymes is required to make ethanol or acetaldehyde convert to acetate, which is a non-toxic chemical compound. And if some people has very weak activity of acetaldehyde, or some people, aldehyde dehydrogenase, or alcohol dehydrogenase, and they tend to be sick, even small amount of alcohol. Seven forms of alcohol dehydrogenase protein so far are found. ADH, alcohol dehydrogenase 1 through 7. And this enzyme, ADH7, expressed in stomach. So male has ADH7 form of this enzyme, but the female, in general, female does not secrete this form of enzyme in stomach. But some Caucasian people has polymorphism of ADH2 form of enzyme. Maybe some mutation, some Caucasian people has, and they cannot convert ethanol to acetaldehyde efficiently. So it says developing alcoholism, tend to develop alcoholism in some Caucasian people. So this enzyme expressed in liver. So most of activity to convert ethanol to acetaldehyde, this enzyme contributes very much, maybe more than 70% or 80%. So only 20 or 30% of ethanol will be converted to acetaldehyde by using ADH7 in stomach in the case of male. So female can utilize only this form of enzyme, ADH2. So I'm discussing about, how to detoxify ethanol, acetaldehyde. The mechanism of breakdown of ethanol or acetaldehyde in this part. So Kenny, can we make 5-minute break here?
Professor, sorry.
Oh, that's okay. Can we make 5-minute break here to move on before we move on to next
Okay,
slide?
Professor, no problem.
Okay.
If 5 hour 2. Yeah, it's okay, Professor.
Okay.
In 5 minutes, ¿no?
Okay. Okay.
Yeah, okay, Professor. Thank you, Professor.
Okay. Thank you.
Chicos, entonces tenemos 5 minutos para poder continuar con nuestra reunión. Son las 5:02. Estaríamos regresando a las 5:07. ¿Sí? Muchísimas gracias, chicos.
Okay, thanks, Professor.
Okay, thank you. Involving.
Professor, sorry, it's okay,
Yeah.
continue.
Oh, okay.
Okay, Professor, thank you, no problem.
Okay, let's start the second part of this afternoon. Okay. So this afternoon, I try to focus on the issue, but alcohol is not very good. So this is the data showing effect of alcohol on our health. So this axis shows number of drinks per day. So I think it's one glass, 200 milliliter. It depends on the alcohol level, but maybe two glass of wine, 3 glass of wine, 4, 5, 6. And this axis shows the relative risk for the life span. No alcohol, one alcohol, risk for cancer slightly decrease compared with people with no alcohol. And the risk for coronary heart disease decrease with 1 drink of alcohol per day. 2 glass of alcohol should be okay, not much problem. The risk for cancer increase even 2 glass of alcohol, 3 glass of alcohol. So more alcohol makes increase of the risk for cancer. So 1 glass should be better than people.
Professor, sorry. Your microphone is in mute, maybe.
Can you hear me?
Yes, Professor.
Oh, sorry about
Yes.
that. Thank you very much.
No problem.
So
Thank you, Professor.
Okay, so this shows the risk of some disease by taking alcohol. So open circle. This is the data of any disease. So this is the data combining many diseases like cancer, heart disease, accident, and violence. Like that. So this axis shows how much alcohol we will take each day. So this data is a risk for disease with no alcohol. This axis shows relative risk. So this should be 1. And if we take more than 3 drinks of alcohol each day, most of disease, except for heart disease, the risk increase compared with people with no alcohol. But 1 drink of alcohol, 2 drinks of alcohol should be okay. About the same or slightly reduce the risk for some disease like cancer or this is a heart disease. But we shouldn't take more than 3 drinks of alcohol. So maybe 600 milliliters of wine. So we had better stop 3 to 400 milliliters of wine each day. And some risk of some disease goes down. Maybe by some psychological effect. I am not sure. But this is the data published by Dr. Marmot in 1991. So situation may be different these days. So still, we shouldn't take lots of alcohol, but some alcohol may have benefit. We have some benefit from taking some level of alcohol. And this afternoon, I try to explain what is the reason why too much alcohol increase the risk of some disease. And I explain female, tend to have the capacity, not efficiently to digest, to degrade alcohol compared with male too. Male does, because of some reason, water level, water amount, or enzyme expression in stomach, or activity of enzyme in liver compared with male. And some Caucasian people has mutation within alcohol dehydrogenase 2 form. They may have some risk to be alcoholism. So next, gene. So so these are discussion about alcohol dehydrogenase gene. Alcohol dehydrogenase gene catalyze ethanol to acetaldehyde. So next, I will discuss about alcohol acetaldehyde dehydrogenase. Acetaldehyde dehydrogenase from acetaldehyde to acetate. This enzyme catalyze. This is very important because acetaldehyde has some toxicity, which can be make mutation. This can be considered as a carcinogenesis to make cancer. And this is the data showing the DNA sequence, some mutation found in gene coding for acetaldehyde dehydrogenase 2 gene. People in Europe, they can degrade alcohol very efficiently. But people in Asia, especially East Asia or Southeast Asia, people do not break down alcohol efficiently. One reason is the mutation found in gene encoding for acetaldehyde dehydrogenase 2 gene. So people with gene ATCTG in the gene of acetaldehyde dehydrogenase gene, ALDH2 gene, they should degrade acetaldehyde efficiently to acetate. But and people in your area, or you can degrade it. You can detoxify acetaldehyde it seems to be very efficiently, just like people in Europe. But people in East Asia or Southeast Asia, we have mutation. More people has, like in Japan or China, people has GCCTG rather than ATCTG. Two mutation. GC instead of AT, we have, people has. So enzyme expressed from this gene, black gene, has low activity to convert acetaldehyde to acetate. So people in Asia are not very strong to digest ethanol, especially acetaldehyde, efficiently, compared with people in Europe, maybe compared with people in your country. And also this red sequence, GCCTA, rather than ATCTG. One mutation, two mutation, three mutation. This red gene has. So people with mutation like GCCTA within the gene coding for ALDH2, more people lives in Asian countries compared with other area in the world. And it means that, people with gene black or red cannot break down, cannot degrade acetaldehyde efficiently compared with people in your country, in Europe. So about half of Asian people has mutation in ALDH2 gene, which cause unpleasant feeling by drinking. This enzyme converts toxic compound of acetaldehyde to non-toxic acetate. But some people with mutation into this gene cannot convert efficiently from toxic compound to non-toxic compound. So two genes are very important to detoxify ethanol efficiently and to have more beneficial effect by ethanol with some psychological effect. So again, about half of people in China and Japan, more than 40% of people in China and Japan has mutation within a gene of ALDH2. And most of mutation makes low activity of this enzyme. Why the acetaldehyde has a toxic effect? Why acetaldehyde makes cancer? So this slide shows the reason why acetaldehyde, this is acetaldehyde, CH3CHO. This attached to the molecule, which is materials for making DNA, deoxyribonucleotide, like, this is a guanosine. So these small compound, acetaldehyde, attached to the materials for making DNA And acetaldehyde modify this molecule. So I put some note. If one molecule of acetaldehyde bind to deoxyguanosine, yeah, and this compound may, if one more acetaldehyde molecule bind to this molecule, and this molecule will be produced. And if this molecule will be reduced, and this type of molecule will be produced. So many different molecules will be produced by binding with acetaldehyde. So these cause cancer. These cause mutation. If these molecules will be utilized to replicate DNA, to make copy of DNA. If these will be utilized instead of this one, and this causes the mutation of gene. And if this mutation occurs in the region of DNA, which promote the cancer production, and this makes increase the risk of cancer. So this is why acetaldehyde with a molecule cause many diseases. So this disease, acetaldehyde produce microcephaly, skin pigmentation, cafe ore spot on skin, limb abnormality on the foot, and short stature. So developmental defect, acetaldehyde, mutant acetaldehyde mix. It means low level of activity of acetaldehyde dehydrogenase because of the mutation within the gene makes many diseases, including bone marrow failure and blood cancer, acute leukemia. So this enzyme is very important. We need to have high level of activity of acetaldehyde dehydrogenase to detoxify acetaldehyde efficiently. But some people, like in Asian people, has very low level of acetaldehyde dehydrogenase to enzyme activity. So they may have high risk of this problem compared with people in other areas. So these people may have high risk to have this disease due to the mutation of acetaldehyde dehydrogenase gene, and taking large amount of alcohol also affect these people severely, because these people cannot detoxify acetaldehyde. Cannot detoxify acetaldehyde efficiently because of the low level of enzyme activity of ALDH. So we can find out if people take only 10 milliliter of beer, and some people, change their color of face pink very rapidly. So this is one signal who has low level of alcohol dehydrogenase 2 gene mutation who has low level of enzyme activity of alcohol dehydrogenase gene product. And alcohol itself also has some effect on our health. We have discussed about how acetaldehyde affect our health. This will bind the materials, raw materials to make DNA, and it promote the mutation, and we may have some disease. So from this slide, I will explain some about the ethanol itself. Recently, people discovered that the ethanol binds to protein on the membrane of our body. Okay, this shows some signal transduction system on our body. And ethanol has some effect analgesic. This means pain, like a painkiller. There's a opioid. Opioid is a famous painkiller, and also another one, cannabinoid, is another painkiller. Analgesic effect they have. So this shows the mechanism how opioid or cannabinoid makes analgesic effect on our body. And these chemicals bind to receptor protein. These are protein, red. So the protein locate within the membrane, plasma membrane, cell membrane. And this makes turn on the signal transduction. And this signal transferred to some other protein, like ion channel. So when these compounds, including opioid, painkiller, bound to receptor protein, the signal will be transferred and the ion channel open, and calcium ion or potassium ion move through the membrane, especially through the channel protein. Ion itself cannot penetrate membrane because these are charged chemicals. Charged chemicals cannot penetrate membrane, a lipid bilayer itself. Ion, potassium, or calcium only can penetrate, only can move through protein. So signal will be transferred to the channel protein and open the channel protein, and the calcium move, potassium move, and the cell will be stimulated or maybe inhibited. And this is a mechanism how analgesic chemicals like opioid makes effect as a painkiller. And alcohol here bind directly to channel protein and open this channel protein and the potassium penetrate through this channel protein. So this is so ethanol has the same effect as the opioid or cannabinoid painkiller. So this is the mechanism why ethanol has the effect of analgesic, because of the effect on channel protein, which other chemicals like opioid or cannabinoid makes. So these all compound affect these transporter for iron or channel protein on iron, calcium, potassium. But the difference is in opioid have a cannabinoid bind to receptor protein first and signal transferred to channel protein. But ethanol will not bind to receptor protein. Ethanol directly bind to channel protein and open it. And it makes similar effect as opioid makes. So this is the mechanism how alcohol, ethanol, has analgesic effect. It means probably ethanol has some problem, makes problem like addiction, alcohol addiction, like people who has addiction against the opioid. It makes a big problem in the US. So we have to be careful. Too much alcohol makes some addiction, some problem. Just like people has addiction against opioid or cannabinoid, because of the mechanism or signal transduction system shared between these chemicals. So this shows the increase of the overdose death rate involving opioid. This is painkiller in the US. So these days, illegal synthetic opioid people purchase illegally, and overdose makes rapid increase of the death by taking illegal synthetic opioid. This makes a big problem in the US. They are addicted. Their addiction to the synthetic opioid. A number of deaths by overdose of opioid. 564,000 in 1 year. Wow, many people. So only 1 year, 564,000 people dead by overdose of opioid, including synthetic opioid in the US. So this is a problem, and we have to be careful that alcohol may have some effect on addiction, just like people had addiction against opioid and cannabinoid. So this is the alcohol and drug use disorder as a share of total disease in 2019. So US has high number, high risk alcohol and drug use disorder within total disease. I don't know this. This is Belarus, I think, just next to Ukraine. This is Belarus has also high risk of overdose of alcohol and drug. And UK, United Kingdom, seems to have high level of, but the US and Belarus are the top country who has high risk by a disorder from alcohol and drug. So this is the alcohol consumption via versus GDP per capita. This axis shows the GDP, gross domestic production in each country. And this axis shows alcohol consumption per capita. And I think your country is just next to the average. So this big circle shows the total number on the earth, in the world. So close to this circle, your country located here. I picked up the country in South American country. So in the middle, about within this area, the country in South America are distributed. And yeah, you have your country located this part about the close to total number in the world. I picked up Northern and Central America country. United States is here. Haiti, Guatemala. And this is, I picked up the Asian country in this graph. China is about next to the total number in the world. Here is Japan. So the level of alcohol consumption about the same. China, Japan. Here's South Korea. They have more. They take more alcohol compared with China and Japan. And country like Islamic country, people do not drink very much or no alcohol. Like Indonesia, they are the largest Islamic country in the world. They have a large number of people, and they are Islamic country, so no alcohol consumption. Saudi Arabia, Indonesia, and Malaysia, I think also Islamic country. They have some alcohol consumption. Turkey is also Islamic country, but still they drink some alcohol. This shows the distribution of alcohol consumption by European countries. They tend to drink more alcohol compared with other areas. Yes, they consume alcohol very much. So this shows the distribution of African countries. Consumption of alcohol and gross domestic production. So they tend to distribute this area. not much alcohol consumption they take. This is the alcohol consumption per capita in the world, each country. So European countries tend to drink more in 2018. But still, even in the European country, their consumption of alcohol slightly decreased. France tend to drink a lot of alcohol, but now 100 years ago, they drink a lot of alcohol, but now about half alcohol level they take compared with 100 years ago. This is Italy. People in Italy, they tend to drink more. They used to drink more, but now they drink not very much compared with the data 100 years ago. And this is alcohol consumption by type of alcohol in France. They drink wine very much. So this is the total amount of alcohol consumption in 2014. But the beer consumption increased slightly compared with data 100 years ago. But still they like wine very much. And this is the similar data in Italy. They also drink wine very much, but still they drink more beer compared with data obtained in 100 years ago. This is the data of the United States. People in the US drink more beer, more spirit compared with some European countries like France and Italy. So this is, I think, period. US people are taking or selling alcohol, making alcohol illegal this period. So they did not officially, they did not drink any alcohol. So this is the data found in Japan. I was surprised that they drink spirit very much compared with beer and wine. Whiskey, I do not know. I will ask Dr. Anderson. He is a professor in the university within Australia. So sometimes I communicated with him and he helps me to make some teaching materials. So we had better take optimal amount of alcohol in a 20 grams of pure alcohol level. This should be okay. In general, this level of alcohol by taking every day will decrease the risk for many diseases. But if we have large amount of alcohol, like 1 liter of beer, too much, and more than 2 glasses of sake, wine, may increase some risk of disease. So I will change my subject to lactic acid fermentation. So I explained ethanol fermentation and molecules, some bad effect of alcohol on our health. So next, I will switch my subject to lactic acid fermentation. Lactic acid fermentation is another fermentation. In the absence of oxygen, pyruvate will be converted to lactic acid within some organism, microorganism, which has enzyme to convert pyruvate from pyruvate to lactic acid. So again, this slide shows in the presence of oxygen, we don't make TCA cycle in the mitochondria. The pathway stopped around this area, here's the pyruvate. And if some organism have certain enzyme, lactic acid will be produced in the absence of oxygen. So this is another type of fermentation. We already discussed about alcohol fermentation. This is lactic acid fermentation. So this slide, I thought I already showed this slide in last week. So we will take a look at lactic acid fermentation. We already know ethanol fermentation by utilizing two enzymes, alcohol dehydrogenase and acetaldehyde dehydrogenase to make ethanol. This is a respiration in the presence of oxygen. So we will take a look at the lactate formation from pyruvate. So this shows the pathway, metabolic pathway from pyruvate to lactate in the absence of oxygen with enzyme called lactic acid dehydrogenase. So if organism has lactic acid dehydrogenase, and if such organism will be in the absence of oxygen, pyruvate will be converted to lactate. So this is the structure of molecule of lactate. This is a pyruvate. So glucose, one molecule of glucose produces two molecules of lactate by lactic acid bacteria. Famous microbiologist Louis Pasteur discovered this bacteria. So we have some species of lactic acid bacteria. So these are a name of taxonomy people use to make fermented milk. And these are a name of taxonomic. These are name of taxonomy of bacteria makes cheese and fermented butter people use for lactobacillus. This is a kind of lactobacillus. Looks like, this is a typical shape of lactobacillus. Lactococcus people also use for making fermented milk. This is a typical shape of lactococcus. And the bifidobacterium, this is another type of bacteria. This is not exactly the lactic acid bacteria, slightly different. It has V or Y shape of bacteria, morphology of V shape or Y shape. So many species of lactic acid bacteria or bifidobacterium has been found. So these are two major lactic bacteria producing lactic acid. Because these bacteria are very useful. People use these bacteria to make some alcohol beverage. So I explain a little details about lactic acid bacteria or bifidobacterium. These produce lactic acid. Only difference, one difference is the amount of lactic acid production. Lactic acid bacteria can produce more than 50% of lactic acid from one molecule of glucose. On the other hand, bifidobacterium can produce less than 50% of lactic acid. So less than half of glucose molecule they can produce, they can convert to lactic acid. And they produce acetic acid. So this is the difference between lactic acid bacteria and bifidobacteria. This is one aspect of the difference between these two. And lactic acid bacteria has some subtype: homofermentative, heterofermentative, facultative, heterofermentative. This is the difference. Lactic acid bacteria can convert more than 50% of glucose to, more than 50% will be converted to lactic acid from glucose. It is called lactic acid bacteria. And these are subtype. Homolactate fermentation produce glucose to 2 molecules of lactate and 2 molecules of energy, ATP, by glycolysis pathway. Another subtype, heterolactate fermentation. This bacteria produce glucose to 1 molecule of lactate and ethanol and CO2 and 1 molecule of ATP. So they consume 1 molecule of ATP to produce 1 molecule of ethanol. So this is two different between these two subspecies. And this facultative heterofermentative bacteria, this bacteria has ability both. So it depends on the environmental condition. Sometimes they produce 2 molecules of lactate, 2 molecules of ATP. In some other condition, environmental condition, they makes only 1 molecule of lactate, 1 molecule of ethanol, and CO2, and 1 molecule of ATP. So this has both activity. The bifidobacterium makes small amount of lactic acid and acetic acid it makes by using 2 molecules of glucose. They produce 2 molecules of lactate and 3 molecules of acetate and 5 molecules of ATP. So it looks very efficient compared with lactic acid bacteria. 2 versus 5 ATP. So bifidobacterium seems to be more efficient in terms of energy production compared with lactic acid bacteria. But you can see by spending 2 molecules of glucose, they can make 2 molecules of lactic acid. So it means less than 50% efficiency they produce lactic acid. So their efficiency to make lactic acid is not very efficient compared with lactic acid bacteria. But in terms of energy production, they are more efficient compared with lactic acid bacteria. So these are two big differences between these two bacteria. Both can produce lactic acid. So bifidobacterium, lactic acid bacteria. Lactic acid bacteria can produce lactate by using pyruvate. So this pathway, actually this pathway is a part of glycolysis. But bifidobacterium has another ability, to produce xylose 5-phosphate, makes acetate. So this is the difference. So this bacteria has both, makes lactate as well as acetate. But lactic acid bacteria can produce lactate only. Or sometimes it makes ethanol from pyruvate. But the bifidobacterium has another function to produce acetate as well as lactate. And here is the extra ATP. This is the reason why they produce more ATP. So if lactic acid can produce, lactic acid bacteria can produce 2 molecules of ATP from 1 molecule of glucose. Bifidobacteria can produce 5 molecules of ATP instead of 4 molecules of ATP from 2 molecules of glucose. So 1 extra ATP will be made by bifidobacterium. This is the ATP, extra ATP, that they can produce from acetyl phosphate to acetate. So Kenny, can I stop here? Can I stop here, this slide, and we will continue in April 16. Is it okay?
Okay, Professor. Don't
Okay.
worry. Thank you for this session.
Thank you very much.
Thank you.
I will send the data and could you make proofreading by Spanish translated by Google?
Okay. Okay. Thank you, Professor.
Thank you.
Okay, thanks, Professor. See you later.
See you.
See you. Thank you
See
very
you.
much. See you,
Thank you.
Professor.
See you.
Thank you, Professor.
Thank
See
you
you
very
next
much.
class.
See you.